Also, what we talk about today will not be on the quiz this weekend. Only halfway through the material we have already, writing quiz questions, so I'm not sure you need more stuff on your quiz. So make a mental note. What we're talking about today is not on your quiz. So we'll separate things in your mind, so hopefully when you put them back together, they go back together nicely. Because when we talk about this thing today, we'll start with non-homologous end joining. It is another example that we can use to fix what we call a double-stranded DNA break. So remember, ionizing radiation strikes the DNA and causes the phospholipid ester bonds to disappear, or fall apart, whatever you want to call it. And we get a double-stranded DNA break. And again, what happens after all double-stranded DNA breaks? Nuclease digestion occurs. And the speed at which this occurs, or the time between breakage and repair, is an important thing to consider with non-homologous end joining. The longer it takes for repair, the more DNA you lose. So if we can get to it really quick, it's not so bad. But after a while, it's going to be a lot sort of worse. And this does create a deletion. So we're going to lose DNA regardless of how fast we fix it. This will always result in a loss of DNA content. With recombination pair, we don't lose anything. We might see a gene conversion because we're using a different DNA molecule to repair things. But here, we're losing something. And there's a choice between having a broken chromosome or a shorter chromosome. Broken chromosome is always better than no chromosome, because losing some information is not as bad as losing all your information. So that's why this is sort of an okay thing, and it's done quite commonly. It's just better to have your whole chromosome, at least most of your chromosome, and maybe not losing all of it. Maybe there's a way to go in and fix it later, but who knows. Again, diploid organisms do this quite a bit. So one homolog is broken, the other homolog is okay. So again, in this weird situation of gene dosage becomes an issue. How big of an issue it is, hard to really say. But we do create a problem. And the other thing that a lot of students sort of struggle with is the idea of the word deletion. Deletion is a frame shift, which is a single nucleotide addition or removal, or actually deletion is removal. But also deletion is a large-scale chromosomal change, a loss of information. So when we think about frame shifts, insertions and deletions, typically the idea of one nucleotide or small changes are important. When we think about duplications, deletions, inversions, and translocations, those large-scale chromosomal changes, the word large is a pretty important thing to differentiate the things. So something you see to do in your mind, the word deletion shows up a lot of places. It's just, are we large scale changes or small scale changes? Basically, with non-homologous end joining, we glue them back together as quickly as possible. That's really all it is. Slap them back together. The faster we do it, the less we lose. But in this particular example here, the underlying nucleotides are the ones that have actually been lost through this process of non-homologous end joining. And here's kind of up, just in this sort of illustration here. 1, 2, 3, 4 and 2/3 codons. It's definitely going to cause a frame shift because it's not a multiple of 3. So how bad is this? Small change, but still might have an effect. Does it affect more than one protein, more than one gene? That's the sort of thing you have to think about. You wonder how much DNA do we lose and how fast is that nuclease activity? There's no way to really say. So there's always going to be some loss of DNA. And because we're not changing anything, this is always going to lead to typically a loss of function. Which is also true for frame shifts. Typically all frame shifts are a loss of function. But getting back to the idea of recombination repair, we want to talk about the idea of gene conversion. Basically, with gene conversion, a dominant allele is changed to a recessive allele or vice versa. It only affects one gene typically. What's called gene conversion, not genes conversions. Some people might confuse this with crossing over. Crossing over changes alleles from one chromosome to the other. But typically crossing over is going to switch multiple genes, so we get more allele differences. This just affects one thing. It's a much smaller scale thing we have to deal with. And we don't care how it was identified, but we know it's possible and it does happen in all organisms. And it basically means you flip something from dominant to recessive or vice versa. And there are three common ways in which this occurs. One is recombination repair, which we'll talk about in a second, or at least review. The other is a problem with mismatch repair. Again, normally mismatch repair should only work when the DNA is hemimethylated, but sometimes it functions outside of its normal window. And when DNA is fully methylated, how do you know which nucleotide is the wrong nucleotide whenever there is a mismatch? And when you do recombination repair, there are potentials for mismatches to be introduced. So it's all about how you fix things. And if you fix things correctly, then you go back the way they're supposed to be, but if you fix it the other way, then you make a mutation. So, not to need to worry about these facts, but mismatch repair with fully methylated DNA can lead to gene conversion, because it's half the time you get it right, half the time you get it wrong. That's just what we're seeing here. So going back, right versus wrong. And you don't know, is the G right or is the T right? Was it a GC base pair or was it an AT base pair there? No way to tell, because we fully methylated our DNA and don't know what is right and what is wrong. Again, with recombination repair, there are places where mismatches could be possible, especially with crossing over being involved. Typically without crossing over, there's not a whole lot of place for mismatches to occur. But if they are going to occur, they would probably occur in this little region here, which you may be able to sort of see that maybe there's a potential here where mismatches to occur. But mismatches are much more likely when we do recombination with crossing over than without. And we don't need to know specifics, too much detail, just that it is a possibility. These are the things that can lead to gene conversions. So hopefully remember Holiday junctions. Which type of recombination repair uses Holiday junctions? No. Just mentioned there's two types of recombination repair. One with something, one without that same thing.
Without crossing over?
So is this with or without crossing over? Holiday junctions.
Without.
Holiday junctions are with crossing over. Sorry. It's okay. Quiz will be this weekend. You'll be exposed to this again. And hopefully we get it right on the quiz. So you get it right on the exam. But holiday junction formation only happens when we have crossing over occurring during mismatch, during recombination repair. And so it's not the recombination repair that is making the gene conversion, it's what happens afterwards. And so typically with, again, crossing over during recombination repair, you're more likely to get mismatches. And because our DNA is fully methylated at this point, how we fix this is maybe potentially going to give us a gene conversion event. And just like with before, it's basically a 50-50 chance of picking the right one to fix or picking the wrong one to fix. The next topic is directly related to fixing double-stranded DNA breaks. And we're trying to actually exploit some natural processes that cells do to do what we call genetic engineering. And CRISPR is the hot thing these days in science. Everybody is trying to use CRISPR to make some type of new thing. Maybe you'll be required to tell me what the acronym stands for. There's no E in CRISPR, so don't spell it like you would the thing you put your fruit and vegetables in the refrigerator. Do you know those are called CRISPR drawers? To keep food crisp. Did you know there's a setting where you can open and close it to affect the humidity? It'll also affect how crisp something is. Because as you retain water, you bloat, and as you lose water, you shrink. Anyways, CRISPR stands for Clustered Regulatory Interspace Short Palindromic Repeats. There's only one really important word here, and that's the word palindrome. Does anybody know what a palindrome is?
Same backwards as forwards.
Yes, and for DNA, it takes on a special meaning. Now, most of you may be familiar with some palindromic words or palindromic phrases. Madam is an example of a palindromic word because it's spelled the same forwards and reversed. Another name that's palindromic is Hannah. There's some other phrases. This one palindrome is forward versus reverse. These are a single strand, and palindrome in a double-stranded DNA sequence doesn't actually look palindromic until you consider that it is a double-stranded molecule. So here's an example of a palindromic DNA sequence. It's the same forward as it is reverse. And Madam and Hannah are both the same forward as they are reverse. But because DNA is double-stranded, reverse means the anti-parallel strand, not the reverse DNA sequence. So these palindromic sites are used quite often in bacteria specifically, is where the system was first developed. And lots of proteins are able to recognize these types of unique sequences. And as you change them, we get different things binding to them. So it's been around, we've known about it for a while, but it wasn't sort of used until recently to be really cool. This is basically a way that we can cut DNA specifically, but typically we're trying to cut some non-us DNA. The whole point of this is to cut DNA. We can co-opt that to do something else that's cool. Now, the two scientists that really revolutionized the idea of CRISPR and using it for genetic engineering are shown here. I don't really know exactly how their names are pronounced, so I will say Carpentier and Dubnon. It could be Dubné. I don't know. Never met her. Don't know her. Not sure. I guess this right and left tells us who is who. But pretty cool. They won the Nobel Prize in Chemistry, whatever year it was. It was 2020 or 2019. So not that long ago. They really, really important in this world of we are sort of now and right now, using CRISPR as a gene editing tool. Now, so far we've been really good at using it for bacteria, but a lot of people want to apply it to gene therapy. Has anybody ever heard of the term gene therapy before? The idea that we could replace a bad DNA sequence with a good DNA sequence should be a way to eliminate disease, if you know exactly what DNA sequence to change. Problem is, the efficiency in changing that DNA sequences has been really low, until CRISPR has increased that possibility. And right now, just so you know, there are a lot of trials that are trying to develop CRISPR technology as a way to treat certain types of diseases. And a lot of these, as you look at this list, are cancer-related things. You don't need to memorize any of these, but you can tell this is a hot thing in the world these days. Now for us, one of the things we want to care about is how this thing works. And CRISPR requires two things: an RNA, which provides targeting or specificity, and the other is a nuclease. So we use an RNA to tell the nuclease where to cut. So it's not just going to cut anywhere, it's going to cut at a certain location. That means CRISPR can be targeted to destroy certain things but not other things. And different bacteria have different types of things. Sometimes they can be directed against double-stranded DNA or single-stranded DNA or even RNA, which is a lot of different possibilities out there. But as we talk about CRISPR, we're mainly going to focus in on the double-stranded DNA aspect of how this works. And so first thing is just to think about how it works naturally in a bacteria. And in bacteria, it's more complicated than in the applied system that we're gonna really learn about. In bacteria, actually 2 RNAs are required rather than a single RNA. One RNA provides the sequence specificity, which is what we call the guide RNA, or called gRNA. The other is called the tracer RNA, and this has 2 important functions. It binds to the guide, but also recruits the nuclease, which we call Cas9. So without this one, there is no recruitment of the nuclease, so no digestion. Without the gRNA, there's no target specificity, so there is no nuclease activity also. And what was the cool thing about what Carpentier and Lobda, I'm not sure if that's differently, but I don't know. You say it, who, what? Is that how it's pronounced? Okay, I don't know. Lobda just, I don't know, just doesn't sound right, but that's the way it comes out, I'm sorry. I'm from Oklahoma, we don't want to talk like me, but apparently that's from the blaming on my heritage. Oklahoma. Sometimes my country does come out, but very rarely does it. I keep it under tight wraps. I don't ever wear country common boots, cowboy boots. Anyways, what those 2 women of science did was to combine the function or the guide RNA and the tracer RNA together in what's called a synthetic guide. So this synthetic guide RNA now has both functions instead of having to deal with 2 separate things. It's both the target specificity portion, but also the Cas9 recruiting function. And the cool thing about this is we can make our own RNA that has a certain sequence that goes with a certain gene. So we can cut DNA at specific places. And that is the real power of CRISPR. Not that it cuts DNA, it's that we can make it cut DNA at specific locations. So we make a double-stranded DNA break. And what happens when you create a double-stranded DNA break?
It's digested.
Nuclease activity happens almost immediately. We start digesting DNA around that area. So this is directly related to recombination repair because we can use recombination repair to make something happen, or we can rely on non-homologous end joining to make something else happen. So remember, with non-homologous end joining, what is the ultimate outcome? Deletions, we lose DNA. So that's one of the things we can do with CRISPR is to delete something, remove it from the chromosome, or remove enough of it that makes a completely non-functional protein. Sometimes useful, something is overacting, take it out, maybe that causes or solves the problem. But really, with gene therapy, one of the more important ideas is can we replace, put something in its place. And to do that, that's where recombination repair is actually used. And recombination repair relies on a template, something to use to replace the missing DNA that was lost. So as we look at what's going to happen here, to create a deletion by using the CRISPR system or the Cas9 system, we're relying on non-homologous end joining to just glue this thing back together after some DNA has been lost. And in this system, there's no template to use to repair the missing DNA. So it gets eaten away, it's lost, and then it gets put back together, and then loss of DNA has happened. You don't need to know anything more about how it works. It's just, again, this is relying on the natural function of non-homologous end joining. But what we've done is target specific things to be deleted. Again, if you want to introduce something, we need to provide a template. So if we make our synthetic guide RNA, we can include a template piece of DNA as well. And you don't need to understand how the reaction works, but if you want to talk about it later and how this sort of goes back together correctly, I'm happy to talk about it more. But same type of system, the RNA binds to the double-stranded DNA, Cas9 cuts the double-stranded DNA, DNA is deleted. Because there's a template DNA around, recombination repair uses the template to fill in what was lost. And here we can insert something we call a replacement type of idea. And nothing too practical, but still, hopefully we understand that to do a deletion, we're relying on non-homologous end joining to do an insertion or a replacement. We're relying on recombination repair, and recombination repair requires a template. So both require the synthetic guide and the Cas9 enzyme, but only one of them requires the template DNA. Really wish I had removed some of these slides so you didn't get to see all this stuff you can do with it, but these are actual studies that have been done. This one was from 2022, so not long ago. This group was trying to make a hyperallergenic cat. See, they allergic to cats, but still like cats. The alternative is to buy a hairless cat. Have you ever tried to buy a hairless cat before? They're super expensive. But now, with CRISPR, we can make hypoallergenic cats. Then maybe we can make hyperallergenic peanuts. So and then gluten, and then wheat. Everything is hypoallergenic. That's the magic of genetic engineering. They haven't actually been successful, but they're close, I guess. Close to getting some cats. Then you gotta breed them, which is the other hard part. You know, so like herding cats is a hard thing to do. I'm sure they're gonna sit there while you poke them with needles a whole lot. There's even this cool thing where, I don't know if you heard about this, you can grow human organs inside other animals, and pig is so far the thing that is used a lot. You might get a pig heart in the future, rather than have to wait for a human heart. So instead of just waiting for somebody to die to get a transplant, you just slaughter a pig and have some bacon, get a new heart. I'm serious, it's done. There are humans that have these organs in them. They're studying them to see how well they last. And it's all based off of using CRISPR to do it. I don't have an update on this, but if you want to Google people with pig hearts, maybe you'll get an update. Yes?
So if you're trying to like replace, like with, do you have to do this like in an embryo so that it replicates the corrected cells?
Those were all done in that mechanism, yes, where they are doing it sort of in the zygote, so there's only one cell to change. And that also gets back to the idea of why gene therapy is really hard to do. How many cells are there in the human body? How many of those cells actually need to be targeted with our replacement? And how do you ensure that all of those cells get it? That's really the problem with gene therapy is coverage. Are you being able to change all of them? If you just miss one, maybe you're just right back to where you were. So nothing really gets fixed. The other is also delivery. How do you get it? I mean, getting it to every cell is different than just introducing it to a cell. How do you ensure that it actually gets into the nucleus before it's maybe degraded into the cytoplasm? All sorts of weird things have to be worked out. Hopefully it will eventually, it'll be pretty cool. But it was actually used on human embryos. There were, I guess it was 5 years ago now, there was 2 Chinese twins that were born. I forget what the problem was. It was something about the parents had AIDS or HIV positive. And they genetic engineered the embryos to be resistant to HIV. Because they broke all sort of human laws to do this. And the guy, I think he lost his license. He's in jail. You can Google it. Google's fun. I like Google. Finds all sorts of fun things with Google. Are they true? Take it with a grain of salt. But do some research. Any other questions about genetic engineering, mad scientist stuff? It's fun. I told you about the pigs, or there were goats. Goats that had been genetically engineered to make silk in milk. I think I said it was cows before, but it's actually goats. And there they were, same thing as you gotta make the goat embryo and grow the goat, and then see, get the goat pregnant or make it lactate, and then see if it doesn't actually make silk. There's like 10% success rate or 3%. It's a pretty big difference. Anyways. Would you like to move on to the next topic? I know everyone wants to move on to the next topic. Does anyone remember what gene expression refers to? Yes. The combined action of transcription and translation, taking what is in DNA and turning it into a functional product that we call a protein. And this is a highly controlled process, which we call regulation. And regulation can happen in a lot of different ways. We're going to start with some easy-ish examples in bacteria to some more complicated examples in eukaryotic organisms. But one of the cool things about regulation is thinking about humans and human development. Remember, we all start as a single zygote. And that single zygote has to divide and go through the process of mitosis until such a point where differentiation takes over. Where that one cell type makes all the different cell types that are in the body. And why does one cell become a heart cell versus a kidney cell? Something that isn't regulation controls. Remember, every single cell in your body, regardless of tissue or organ, has the exact same DNA. It's just they use it in different ways. Some cells will highly transcribe other things, others will highly transcribe other things. What we call highly transcribed DNA, highly expressed DNA. What was that term to describe the type of DNA in a chromosome that is highly transcribed or highly expressed? Because it has lots of genes in it. Yes, euchromatin. Remember, when we organize the nucleus, euchromatin is typically in the middle, heterochromatin is typically on the outside, but also things the cell needs a whole lot of typically go on the inside of the nucleus. Things that the cell doesn't need a whole lot will go on the outside of the nucleus. If every cell in your body is doing something different with its DNA, does that also mean that the nucleus is organized differently for different cell types?
I think so.
Possibly. I don't know if it's actually true because I haven't looked it up. But I would imagine it would be different. Your heart cells, and DNA in your heart cells might be different than how the DNA is organized in other cells. Because certain things are needed more than other things. So turning things off and turning things on is a really important thing about controlling when cells do things and make sure they don't do things when they're not supposed to do them. Again, I like to show some primary literature every once in a while, and here's just an example of where expression issues of a certain type of DNA causes disease. I don't know what SPON is or SPOAN, but apparently it's this spastic paraplegia, optic atrophy, and neuropathy. Sounds like it affects your ability to see in your brain and your whole body because apparently causes all sorts of problems. Just the simple overexpression of a small DNA sequence can be quite detrimental. So we really need to keep a tight control on things. And when we think about regulation, there are lots of different aspects that can be controlled. And one of the more important ones that we'll talk about is controlling when transcription happens. One of the other ideas that we'll deal with quite a bit is how long does that RNA last inside the cell? Remember, the RNA that is made from transcription is used for translation. So if we stop transcription but the RNA is still around, more of that protein still gets made. So as we talk about regulation, there are other aspects that we need to sort of think about after the fact. If we stop transcription and get rid of the RNAs, maybe there's still proteins out there that are still functioning. What can we do about those? Are there ways to turn those off? Or just we have to wait for them to die out and get old and run down? So there are ways to do these things. Most of what we know about regulation is by studying bacteria. And a lot of what bacteria do is fairly simple and straightforward. But in all types of organisms, there are different levels of expression patterns that we need to think about. And one of those is called constitutive. You may call it constitutive, if it helps you spell it correctly. But it means that we are always on. The gene is always transcribed, and most oftentimes, constitutive also means high levels of expression. This is something we always need, and we always need a lot of it. So we're always making it, and it's always around. And genes that are under constitutive expression typically are what we call essential, or fall in the category of what we call housekeeping. And housekeeping is generally thought to be things you have to do all the time. You can't just stop doing it, because shit goes bad when you stop sort of cleaning and digging up and staying organized. And I'm not giving you any examples of things that would be housekeeping genes, but something you always need. Anybody have a thought? What is a protein that a cell might always need? Any ideas?
RNA polymerase.
Possibly. RNA polymerase. That's not a single protein, but it is a multi-subunit thing, but yes. That would be an example of something you would always need if you aren't actually transcribing one thing, you're probably transcribing something else. Anybody else have any other thoughts on other proteins or things that are made with proteins or of proteins? Some people think ribosomes. We don't talk a whole lot about ribosomes in this class. Ribosomes are made of proteins and RNAs. If you stop making RNAs and stop making proteins, then you don't have anything to make your ribosome. That would sort of suck. So there are certain things that you need sort of all the time, regardless of condition. And constitutive means that we always make the thing at a constant high level. For everything else, there's a way to turn something on or turn something off. We have 3 mechanisms to turn off and on things, which we call repression, induction, and activation. Repression is the one that's different than the other 2 because of its outcome. Activation is the one that's different than the other 2 because of its method. And so we do see similarities between how repression and induction work, but repression and induction will give a different outcome. So there's a lot of confusion between on induction, because it kind of works like repression, but gives a result that's like activation. And we'll try to build this out as we go along. Repression, induction, activation allows us to change expression levels to either go up or go down. And generally, when this classroom uses basic terminology, that we always have what's called an unregulated level, or a baseline level, which we have a certain name. It's not any of these words, it's a different word. That'll show up on the next couple of slides. Then there is the regulated level. So you're normally expressing genes, and then you repress it. That's your regulation you're doing. Repression is the regulation, the change that you are making. Induction is the change you're making. Activation is the change you are making. Now, one of the cool things about these is these are most common in bacteria, and DNA binding proteins are essential for all 3 of these activities. Things binding to DNA and helping or hurting the transcription process in one way or another. As a starting point, it's important to understand that repression means we turn things off. And so if we turn things off, it means they're on to start with. Typically that means they're high to start with. When we turn them off, we decrease the amount of gene expression that we see. For induction and activation, we are doing the opposite. We start out at a low level, and then the induction or activation occurs, and gene expression increases. So for these 2, we go from low to high, or turn it on. This one, we go the opposite direction, turn it off, and decrease the amount of expression that we see. It won't be important for this class, but other classes you may talk about certain types of genes are controlled by different types of mechanisms more often. And just so you have this in the back of your mind for later, catabolic, or genes that are involved in catabolic processes, things that make energy, tend to be activated, whereas things that are more biosynthetic or consume energy tend to be more repressed. And induction is a weird sort of French case, it's kind of both. But when we think about an unregulated level, the most common type of unregulated level that we talk about is called basal. Which base level, base level or baseline, that's where this thing comes from. And a lot of people think basal means low. And sometimes basal does mean low, but sometimes basal means high. Basal just means that's the default state. That could be a high level expression or it could be a low level expression. But typically when we think about basal transcription or basal expression, one of the most important things is not is it high or is it low, but the fact that it's a constant level. It's an unchanged expression level. So over time, there's really no difference in the basal transcription level. For repression, basal is always high. For induction and activation, basal is always low. Because again, one's high to start with, the others two are low to start with. Sometimes we see something called stochastic expression. And stochastic expression is a very weird type of activity. On average, stochastic expression will give a constant level of protein. But when we typically think about stochastic expression, it'll be something that looks more like this. Where there is a burst of expression, and then it's turned off very quickly. It's like spiking something. You get a short burst, and you wait to see how long does it take for it to go away. As soon as it goes away, then you can hit with another spike. But you don't need it all the time, but you need a little bit. So different things are just controlled by different mechanisms. And when we think about basal, constant is the idea, but stochastic is hit it really hard and let it reverberate or dissipate over time. I mean, like music, to play a constant note, it's like a long note, versus hitting the drum, you just make a sound and it goes away eventually over time. But mostly we like to talk about basal expression. It's nice and easy to think about. Okay. So again, repression starts high, goes low, after because of repression. We have a basal level, and then we have repressed level is a low level. For induction, we have a low level, then we induce, and it goes up. And for activation, we have a low level, and we activate, and it goes up. Induce something typically means to make something happen, right? Most common way we use this phrase like induce labor, so to make it happen because we're waiting to can't wait longer, like let's make it happen, right? I want my kid to be born on this day, so let's induce labor on that day. Does anybody know anybody that's done that? I mean, I've known people that have induced labor on certain days, so they have birthdays on certain days. Seems pretty selfish, but what can you do? What's more selfish? Picking a kid's birthday, or making sure your kids don't have the same birthday? But look, there's a protein that's going to bind DNA. And with oppression, binding DNA is going to be the trigger that allows us to do the thing we do. For activation, binding DNA is the trigger that allows us to do the thing we do. For induction, we're no longer binding DNA, we're actually letting go of DNA. So repression and activation both work because of a protein binding DNA and causing the change to occur. Induction is the one that is opposite. Induction and activation both go from low to high, so they're the same. Again, how we do things is a little bit different. And just so you don't forget which direction things go, there's this nice little slide here that does the best I can to remind you what does repression, activation, and induction do. Maybe I should start, I should say basal, and end, maybe regulated. Hopefully we all figure it out. Do we have any general questions about which direction things go? because that is one of the more important things. It's just gonna get more complicated as we go along. Now, before we go forward, we need to go backwards. So let's go backwards a little bit. And I don't remember when we talked about this, but it was sometime during exam 1. We need to revisit that basic idea from earlier. Double-stranded piece of DNA, anti-parallel strands. Somewhere in this DNA sequence is this thing that we call a gene. Upstream of a gene is a piece of DNA. Does anybody remember what that piece of DNA is called? It's important for transcription. That's what we call motor. It's really important for gene expression. We don't need to know the other part because it's really not that important for gene expression, but at the end, what we call downstream, is this piece of DNA that we call the terminator. For transcription to happen, the simplest view is that RNA polymerase, the enzyme that does transcription, has to bind to the DNA around the promoter. And this binding is what starts the transcription process. So as we think about repression, induction, activation, we're trying to do something that's going to affect the RNA polymerase's ability to bind to DNA. And by preventing or helping this binding, we're stopping or starting the transcription process. For a few of these things, there is another piece of DNA that we're not really going to talk about, but really important. So why am I mentioning it? I don't know. I can't not mention it, even though you won't be asked to remember it. There's another piece of DNA called an operator sequence. And the reason why I bring this up is this is typically a binding site for DNA binding proteins. Binding, BD. DNA binding protein site, sure. So as we think about how these things are sort of affecting transcription, they're not binding to the promoter because that's what the RNA polymerase binds to. They're typically binding to another site. The operator site is typically going to be involved in one particular type of activity. The other sort of thing we need to think about in general is oftentimes sometimes there's another site up here called an activator site. So these pieces of DNA near the promoter that are where these DNA binding proteins are going to bind, which is going to affect this thing from getting where it needs to get. And we don't need to know all the details of this idea, but sometimes a little bit more context helps you understand sort of what we're talking about. We're talking about proteins getting in the way, stopping transcription, or proteins getting out of the way to allow transcription, or proteins that are helping RNA polymerase do transcription. Those are the three things we actually are going to see. Get in the way to stop something, get out of the way to allow something to happen, or get nearby to assist. And the first one that we talked about is repression, which is putting something in the way, or induction, we're going to move something out of the way. For activation, we're going to get nearby. We need a partner. Activation is basically the partner is needed to help these things happen. Actually, I'm not going to talk about repression today because that's going to introduce a whole lot of worms. That is not really worth introducing today. But there is one more general idea that I want you to think about. Why is basal high versus why is basal low? Does anybody have any general thoughts on why the base level could be high or low? much.
Wouldn't it be high if it's something we need most of the time, and then occasionally we don't need it, and then low if we don't need it most of the time, as long as we do need it?
Yes, but what makes it high? Not what things would be high. What actually makes it high? And what makes it low? It's much more simple than that. The answer's on the board. What makes it high and what makes it low is what makes it. And what makes it happen, what makes transcription possible? Promoter. Promoter sequence is the thing that where we start transcription. And so, even though you don't need to know this for this class, it can be very helpful in things like molecular biology. When the basal level is high, typically we have a very good promoter. A very good promoter. Which means all the things that are needed for transcription bind really well and where everything works really good. But typically when basal is low, it could be, for one reason, is because we have a very bad promoter. Like in the case of activation, there's nothing in the way, but it's always low. So what makes it low is the promoter is just not a good promoter. That's why we need help. So if you don't need to know this, it's good to have this idea of, you know, the promoter and the gene. Just when we talk about gene expression regulation, you have an idea of where these DNA sequences are located relative to each other, because we're going to see some pictures. And the other thing I just want to mention before we go forward, is last year or the year before, somebody told me that these things don't show up when they look at them on Canvas.
Yeah.
So, do whatever you need to do. Usually when you download those slides, they show up. But when you look at them online through Canvas, they don't show up. And it confused somebody. It was like, where are these things you're talking about? They're right there, don't you see them? So please beware, there are, I don't know what the problem is with these things.